I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and I am excited as usual to bring you this episode, number 126 in this case, in our ongoing effort to provide you do-it-yourself tools for the betterment of your very own brain. This week we're going to be talking specifically about an adaptogen plant called Bacopa. Bacopa Monnieri is the full scientific name. It's both a pretty plant and its extract is a fairly well-known dietary supplement and has some really significant cognitive benefits well worth knowing about. I'm going to be speaking in the main interview with Professor Con Stough from Swinburne University in Australia. Dr. Stough is a frequent interview guest on Smart Drug Smarts. This is now his third time on the show, and we'll be lucky to be speaking to him once again today. That'll be in the main interview. If you hang around until the very end of the episode, touching back to last week's episode, which was about exercise and cognition, I'm going to tell you about some recent findings as to the best form of exercise for inspiring neurogenesis in a particular region of the brain. That'll be in the ruthless listener retention gimmick. But as usual, let's kick things off first with This Week in Neuroscience.
Smart Drug Smarts. This Week in Neuroscience.
So maybe some of you have had this experience where you've spent a significant amount of time in a country overseas where you don't speak the language or you don't speak the language very well. And then you come back home and you find yourself having conversations with pretty much anybody that you can get your hands on. People like the grocery store clerk or the mailman or people that you just don't really know because you've just been dying for the social interaction. And if this is how you feel after a trip, imagine what it felt like to be like an Old West gold prospector that's alone with your mule in the hills for weeks or months at a time. And then you stumble back into town covered in dust and you're just dying for conversation with anybody. Well, this sort of socialization rebound that we know pretty well to expect in humans, it turns out that this occurs in mice as well, which is kind of interesting unto itself, but what's even cooler is that they've figured out an actual patch of neurons that seems to control this effect. This discovery has just taken place and was reported in the February edition of Cell magazine by scientists at MIT. But this area discovered, again, this is in mice, called the dorsal raphe nucleus, or the DRN, appears to be responsible for feelings of loneliness and to mediate the social bounce-back effect when mice are reintroduced to environments with other mice. Says Kay Tai, one of the lead researchers in the study. To our knowledge, this is the first time anyone has pinned down a loneliness-like state to a cellular substrate. Now we have a starting point to really start studying this. The identification of the DRN as being related to feelings of loneliness in the mice was a bit of lucky serendipity. There was actually a study going on as to how the DRN would respond to drugs, and the mice in the control group had to be isolated from their peers for 24 hours, and researchers discovered that lo and behold, there was a strengthening of connections in the DRN in the control mice during this period. They didn't expect to see any changes in the control mice, so that made them suspect that something was going on, and they started looking at this further. A secondary round of testing trying to figure out what was going on here, they used optogenetic controls to suppress DRN activity in mice that had been isolated from their peers, and then when those mice were reintroduced to other mice, those mice that had had the DRN suppressed showed less excitement, less of this social bounce-back behavior when they were put back with their friends. So this would be akin to the old mining prospector coming back into town and not really being particularly enthusiastic that there were people to talk to. There are also other interesting mediating factors, like the amount of social dominance that a particular mouse had within the group definitely seemed to affect the social bounce-back response. So if you're a socially dominant mouse, let's say Mickey or Minnie or something like that, and you're put back with your peers, you're going to party quite a bit more in mouse terms than if you're a low-ranking mouse on the mouse totem pole. And that kind of makes sense. The socially dominant mouse has more to gain by interaction with its peers than a socially less dominant mouse. The next study that the MIT researchers have planned is to find out whether these neurons are detecting loneliness or if they're also responsible for driving the response to loneliness. This seems hugely interesting, and it makes you wonder what sort of therapies could come out of this. There's that Jim Carrey movie from about 10 years ago, The Eternal Sunshine of the Spotless Mind, and I could think of a scenario like that where some people might find loneliness to be so debilitating and demoralizing that they might want to just zap this part of the brain and take it offline if it does turn out to be the sensor on the loneliness alarm. Not that that's necessarily a good idea, but it sure is intriguing things like that might even be possible. The scientists have not yet looked for and found the analogous spot within the human brain, but they do sound pretty confident that this would be a common feature to mammal brains, and they will find something analogous within humans once they look.
Smart Drug Smarts, the podcast where smart people talk about smart drugs.
Our interview guest this week is Australian, and I figured I would check out the Australian iTunes reviews. Had a couple of 5-star reviews down there. Marty the Mud Crab said, "This is without a doubt my favorite podcast. After discovering it, I burned through nearly 100 episodes in about 2 weeks. Now that I'm hooked, a week between each episode seems like an eternity." Sorry about that, Marty. That is amazing. 100 episodes in 2 weeks. I hope you got something else done during that time too, but very flattering. And this is actually weird. The other Australian review is from Toby the Cow. So we got a mud crab and a cow. Toby says, "I'm not a person who gets stuck into podcasts easily, but SDS is really informative, enjoyable, and well produced. I enjoy listening to this while I walk, and now I walk twice a day just to enjoy the podcast." That is excellent to hear. Walking is one of these things, by the way, that just keeps getting more and more good publicity. I think I've already mentioned on this show a book that I read last year called Get Up, Why Your Chair is Killing You. This was a really, really good popular science book. It's got science, it's got research, it's got stuff you can take action on, and it's also just a really well-written, entertaining book. Not at all dry or stuffed shirt or anything like that. I'll make sure that we have that up on the smartdrugsmarts.com/bookshelf page. But not surprisingly, Get Up talks a lot about walking and its benefits, and the closely related concept of NEAT, that's non-exercise active thermogenesis, which we touched on a little in last week's episode, but is probably the single biggest weapon any of us have in the arsenal for overall physical health and maintenance of a healthy weight, avoidance of cardiovascular issues, and all without major overt effort. The effort with NEAT is never about physical strain, it's more about making it work with your lifestyle and your schedule, just finding opportunities for physical movement throughout the day. I'll cut myself off on that rant now, but one place where you will find me ranting with relative impunity is the Smart Drug Smarts newsletter. We call that the Brain Breakfast. And if you're not signed up for that yet, let me invite you over to smartdrugsmarts.com/newsletter, where you can very quickly do so. We'll keep you up to date on all the stuff that we got going on. A couple of fun things coming up this summer. I'm starting to get excited, I must admit, about the week-long water fast that we're going to be doing at the start of June. Sort of a mixture of excitement and trepidation, much as it was last year, but at least this time I know what I'm getting into. Anyway, jump on the newsletter to get early warnings about all that stuff. I will also take this opportunity to tout axonlabs.io. That is our retail home on the web, where you will find a couple of stacks that we released last year, Nexus and Mitogen, both of which, as you would expect, I am a huge fan of. I got an email this past week from somebody that said Mitogen has been absolutely instrumental for her, lifting brain fog that she'd had a hard time getting out from under ever since she'd contracted Lyme's disease. Mitogen is a mitochondrial enhancer. Mitochondria, as you know, are the powerhouses within every cell in your body, and it's nothing that I would have thought of prior to getting that email as having anything to do with Lyme's disease or the related symptoms, but the general utility of mitochondria throughout your body, and particularly in the brain where they are densely populated. I can't say that I was too surprised. It was great to hear that it was helping her shake off that brain fog. Mitogen and its sibling stack Nexus are both available on the web over at axonlabs.io, or you can visit smartdrugsmarts.com and click over from there. I think that's all the housekeeping that we have for this week, so let's move along now to the main interview.
Smart Drug Smarts.
So if you've been listening to Smart Drug Smarts for a while, Dr. Con Stough will be a familiar voice. He is a professor of cognitive neuroscience and psychology at Swinburne University in Australia, as well as the director at the Center for Human Psychopharmacology there. He's got a wide range of research interests, everything from theory of intelligence to natural medicines to drugs and driving, all over the place. But one particular area of interest for him is the adaptogen plants. An adaptogen, again, we define as any natural herbal substance that is supposed to help the body adapt to stress and to exert a normalizing effect upon bodily functions. Bacopa, it turns out, has a lot of distinctly cognitive effects, which makes it of particular interest to us, and that's what we're going to be talking about with Dr. Stough in the upcoming interview. Bacopa is also a very attractive plant, cute little white flowers. Sometimes we'll be pulling up images of the things that we're talking about on this show, and if it's a synthetic compound, it's just a pile of white powder, which is never particularly photogenic. But Bacopa is a different story, so if you get a chance to head over to the Smart Drug Smarts website, take a look at that. You will not be disappointed. But let's move to the interview now with Professor Con Stough.
Bacopa is probably the oldest known herbal extract. It dates back into ancient Indian scriptures or books, more than 4,000 years. And it has been used very widely in that period of time as a sort of anti-inflammatory, as a medicine for children to increase memory and cognitive capacity. Mm-hmm. So the historical use really parallels the more sort of modern use. And of course, extracts have become more sophisticated since that time. But I think it is the oldest known herbal medicine.
That's really interesting that even early in history, it was recognized this was a cognitive enhancing compound in young people.
Yes, that's right. And I don't exactly know the context, obviously, going back all that time, what other problems in society, young children faced. But certainly it's still being used for kids with the cognitive or even behavioral problems in India. And we're currently running a trial with kids with very high hyperactivity and inattention, which sort of parallels that use at the moment. We're about 2/3 of the way through that study and should be finished by October, we hope.
Does the plant naturally grow outside of the Indian region? Is it in other parts of the world as well, or is it pretty localized?
I'm not really an expert on that area, but I believe that it does grow in other parts of the world as well. I was talking to a botanist last week. There's different versions of Bacopa grown in other countries, even in Australia, in Tasmania, for instance. So probably quite similar compositions in terms of the active components and those sort of things. But obviously it must have some sort of specialty derived from India, and people have historically used that in India. And if you go to other countries, they've used other herbal medicines. So there must be some specificity for India.
Nowadays, when people get Bacopa, it's typically coming in a capsule form. But what part of the plant is it coming from originally, and how was it historically used in either foods or extracts?
I think like all herbal medicines, so people have experimented with different parts of the plant, but I think the more modern day ones have been a sort of flowering plant extract. Ginkgo, for instance, people have not only used the leaves, but also like the roots and other parts as well, with varying success, probably for different indications. But I believe Bacopa extracts come from the flowering parts of the plant, which is, that sort of white flower. And it's an aquatic herb, so it grows around water. And there's been, reports that it grows on the banks of the Ganges and things like this. And that sort of gives us a bit of a clue about how it works. It sort of filters through pollutants and toxins that are found in that part of the world.
What are the effects that we've seen on human cognition from modern testing, not just from historical hand-me-down wisdom?
This is really interesting. It has so many different effects. So different than a pharmaceutical, which has a very strong single domain. And many plants in this space are what we call sort of polypharmacological. That is, they affect many different areas of our biology, particularly brain function, at the same time. So there's been a huge amount of research, actually, particularly in India. Firstly, in vitro and animal research. The Indian government has done an amazing job over the last 50 or 60 years to publish hundreds and hundreds of scientific papers that try to identify the mechanisms in vitro and animal research. And there's a government agency called CDRI, which is the Central Drug Research Institute in India, which is a government-funded institute that have spent a lot of time doing really great research on this topic. And they've typically found so many different things. So like many plants, it's a very strong antioxidant. Unlike many plants, it also has some very unique mechanisms. Some studies have shown that it affects something called beta amyloid, which is a protein that has been linked to
Alzheimer's.
Dementia, exactly, yes. And it also has very strong specific effects on inflammation. And again, as we get older and in patients with dementia, they have an unusual amount of increased brain inflammation, which we're worried about. And so some of the Bacopa extracts have shown to have very specific effects on some of the interleukins, for instance, IL-1, 6 and 10, and very strong anti-inflammatory effects. Some studies have also shown that it might be able to chelate some heavy metals in the brain. So lots of really interesting things that might improve cognitive function. Seems to also have some positive effects on the cardiovascular system, increased blood flow, for instance. And a really interesting study a few years ago showed that it increased synaptogenesis at the same time as improved learning in animals. And so if you take all these mechanisms together, really does seem to be a really promising extract for brain function and cognitive function, particularly as we get older.
Are there any contraindications, or are there people that are allergic, for example, to Bacopa? Is there somebody that might want to avoid this for any reason?
Well, look, you never know, do you? I mean, some people can react to absolutely anything, so it's a bit of a mystery sometimes, but it's very, very well tolerated. And a lot of these plant-based extracts have been used for thousands of years. The most common side effects, some gastrointestinal issues, but they usually go away after a few days. And it's something that you want to take with food, we generally recommend. And in our clinical trials, we administer it with a bit of food. And also because it might have some direct cholinergic effects. Some of the studies have shown that. And we recommend that people don't take it at nighttime because Obviously, there's a relationship between acetylcholine and sleep as well. So sometimes it can keep people up if they take it too late. Not all people, but some people. So we suggest take it with a bit of food in the morning or at lunchtime. And generally, the few percent of people who had wine in Australia, there's hundreds and hundreds of different Shirazes grown in Australia. And they're all grown differently and they're extracted differently. And you can buy a bottle of Shiraz at the supermarket for $2. Or you can buy a bottle of Grange Hermitage, which costs you $600. You know, they're grown in different places in Australia in different conditions. They're extracted differently. They're treated differently and they taste differently. And it's the same thing with herbal medicines. The really high quality ones are grown in the same places. We know where they come from. There's a really complicated high science extraction methods in which we know from batch to batch what's contained in terms of the active components in the capsules that you're taking. And unfortunately, price and quality are often correlated in this area. And you can buy certainly cheap herbal medicines from the pharmacy or the supermarket. But when people have gone to do proper analyses of the chemical compositions in them, they find that they vary hugely. And they're different than the ones that have been used in high quality research. Right. So you can buy something which have almost every chance of not working from the pharmacy or from the supermarket, or you can buy a medicine that has been proven to be consistently made and to have high efficacy. So this is really a message we really need to get out to the consumers. And it's really important for Bacopa because the research that we've based a lot of our work on comes out of that scientific government-funded lab in India called CDRI. And they have published literally hundreds of publications on this particular extract, which they call CDRI-08. That's the scientific number for it. And we know about all the mechanisms that it affects. And then we've gone to do human clinical trials with that same substance. So people really need to try to find that extract or a similar extract that also has high quality. So I don't want to really plug any companies, but just as an example, that extract that we use is called in Australia and most parts of the world is called Keen Mind. It's a sort of commercial name. And I think in the US it's called Synapsia. And there may be some other high quality extracts as well, but there'll also be a lot of really low quality extracts as well. So people need to be pretty careful about which one they choose.
In a way, I mean, it's nice if the quality and the price at least correlate. I mean, the worst case scenario is to pay a lot for a high-priced product only to have it be as bad as the low-quality product.
Yeah, well, that's right too. But the other thing about a low-quality product is that they might have a lot of impurities. So that's a double whammy, isn't it?
So assuming that people are finding the right product, a high-quality extract, what sort of dosage might somebody be looking for on a day-to-day basis?
Yeah, so typically first you want to look at what's in the Bacopa. So the one we tend to do research on has got about 55% bacosides. So the bacosides are thought to be the important active ingredient in the substance. And then once you've got about that level of bacosides in the extract, you need around 300 milligrams seems to be a good dose for adults. And traditionally about half of that has been used in children, although there doesn't really seem to be any real reason for that. It's more of a historical thing. But in adults, it looks like 300 milligrams seems to be pretty efficacious.
And you mentioned that taking it with a little bit of food is generally a good idea. You know, some things are lipophilic versus hydrophilic. Should there be some fats in the food or does that not matter?
Probably helpful, yes.
One of the questions that I've had presented to me, and I'm sure you've heard as well, some of these adaptogens almost sound interchangeable with one another as far as the benefits. When would I choose something like Bacopa over something like rhodiola or one of the other plant-based compounds?
Yeah, I think rhodiola is quite a good product as well. We've had some pretty good data on that. Improvements in anxiety and feelings of well-being. But I don't think there's a big literature on its cognitive enhancing effects. So I would definitely be choosing Bacopa, a good extract of Bacopa, over anything else that I know of. If you're looking to improve cognition chronically, particularly as you're getting older, I think all the sort of pharmacological effects of Bacopa seems to map on really nicely to age-related cognitive decline and the biological mechanisms that are related to brain aging, changes in oxidative stress, changes in inflammation, decreased synaptogenesis, metal chelation, beta amyloid changes. seem to be related to brain changes as we get older. You could probably take rhodiola and Bacopa together. I think they would have quite complementary effects.
Is there anything else that seems like it might be a good complementary mix with Bacopa?
Well, that's the thing. Companies are now starting to ask that question. Can we design nutraceuticals now that Bacopa plus something else? It's really depending on the indication, I think. Bacopa has some very specific properties. You might then look at other molecules that have different types of properties as well, thinking, well, will these things have complementary effects? And we don't know because, in fact, Bacopa probably has 50 different active components in it, much like ginkgo. And what happens when you start putting these things together? Do they help or do they hinder each other? I think we're at the beginning of that sort of research question. I think that the research is really showing consistent cognitive benefits. There's now something like 10 or 11 chronic randomized clinical trials with Bacopa extracts, and everyone except for one is showing a positive benefit. So this is really something that's looking quite consistent and positive, and something which we really need to start to sort of ramp up the larger trials. And the trials that we really want to do are the sort of preventative trials for dementia now. Taking people who are elderly and giving them high quality Bacopa extracts, and maybe even combining that with cognitive training, for like 4 or 5 years and seeing how many convert to dementia. That's really where the research needs to head to.
You mentioned earlier synaptogenesis is one of the benefits that can come from Bacopa. A lot of people are familiar with the term neurogenesis, but synaptogenesis might be a new one. It's a different but related idea. Can you kind of break apart those two and talk about the differences?
Neurogenesis is sort of an interesting concept. It's sort of been a bit of a holy grail. So it's the idea that we can generate new neurons, new brain cells. And for a long time, we thought that at a certain age, we couldn't do that. And we're starting to rethink that, although I don't think we've really come up with great ways of doing that yet. Some of my colleagues are playing around with things like fish oils and omega-3s, thinking that might improve neurogenesis in, say, the hippocampus, for instance. And I know people are trying to develop pharmaceuticals that try to get the brain to generate new neurons. But synaptogenesis is a little bit different. Synaptogenesis really refers to getting the brain to make new connections with itself. As we get older, the concept of use it or lose it might refer to this idea of synaptogenesis. So the idea that we make connections, every time we learn, we have to make new connections in the brain. There has to be a biological representation of new memories and new learning and those sorts of things. So people have heard of cognitive training, for instance. There's some, very popular cognitive training platforms that are advertised quite a lot. And some of them are starting to show some success, particularly as we get older. Is the idea that we might force older people to learn new material, things that they haven't learned for a long time, and that might increase synaptogenesis. There was a really interesting study done a few years back that's got animals to learn to do a learning task, and they essentially gave them either placebo or Bacopa. And then something which we can't do in humans, they can do in animals, they can then open up their brains and count the density of neurons and the density of connections in the brain. And they found that the animals who were treated with Bacopa, not only did the learning task better, but they had more connections and they had denser interconnectivities. So they had increased synaptogenesis. So we're thinking that Bacopa plus new learning, cognitive training could be a massive step forward in terms of getting the brain to make new connections and increase synaptogenesis. And as we get older, maybe as we get lazy or something like that, the brain undergoes neural pruning and we lose these connections in the brain. Perhaps we're saying to the brain, we don't need these connections any longer. And, maybe that's the basis of, older people having quite sort of rigid thoughts and inability to be interested in new learnings. So that's something we really have to attack, I think, trying to help older people generate these new connections.
Yeah, and sounds like just one more vote in favor of people constantly looking for opportunities to step outside their comfort zone, expose themselves to new skills, new ideas, and stuff that's hard. It just seems like there's never anything bad to say about that.
Yeah, I think so. And when I give talks and older people say, well, it's great that I'm doing crosswords or I'm doing Sudoku. But I often say to them, look, if you've been doing that for a long time, you're really comfortable doing that. Try doing something that you're not comfortable with. Try learning something which is really hard for you, whether that's a language instrument or a different cognitive task. As you say, it's really important to get out of our comfort zone in terms of learning.
Smart Drug Smarts.
So thank you so very much for Professor Stough taking the time for that interview. Really important points there about not all extracts being created equally. I know that a lot of people feel positively inclined towards natural products versus synthetic products and opting for things when possible that come from nature. And I certainly understand that sentiment. There are pros and cons. One of the downsides of the natural products from living things is the variability based on the extraction method and the particular genetics of the plant or the fungus or whatever it is in question, as well as the fact that, as Professor Stough mentioned, there can be so many bioactive constituents in one of these extracts that there's just a whole lot of different things going on. There could be different mechanisms of action. Somebody could be allergic to one compound within a plant, not allergic to another compound. The complexity just really balloons when you're talking about something derived from a living thing. But the key takeaway being, when we're looking at studies that show results from natural compounds, particularly results that we want to achieve ourselves, paying particular attention to things like the extraction method used in those tests, and then making sure that actually is in sync with what we're getting in our supplements is obviously crucially important. We'll put a link up to Dr. Stough's recommended providers at smartdrugsmarts.com/126. And now, as promised in the Ruthless Listener Retention Gimmick, a little tidbit of helpful advice if you're plotting out your workout and you want to tip the odds in your favor of having just a bit more neurogenesis going on within your brain. Talking about recent studies related to that is what we got coming up next.
Smart Drug Smarts, Ruthless Listener Retention Gimmick.
So last week we talked about exercise and the brain, and I wanted to touch on that just one more time with a recently published research paper on neurogenesis, that's the growth of new neurons, in adult animals, adult mice in this case, and in that famous area of the brain, at least famous as brain areas go, called the hippocampus, which is, among other things, very important for the formation of memories. And the hippocampus is also an area where they've got more of a slam dunk case for the growth of new neurons throughout the lifetime. We do know now, sigh of relief, that adult neurogenesis does happen in humans and other mammals, but it does not seem to take place in equal amounts throughout the brain. And the hippocampus does seem to be one particularly gifted area in the growth of new neurons in adult animals. So then the question is, what type of exercise is best for stimulating this hippocampal neuron growth? And according to this study, in rodents at least, it is aerobic exercise, such as running, but this probably translates to cardio workouts in general, that seems to have the most effect in promoting adult hippocampal neurogenesis. They also tested these mice with high-intensity interval training, as well as anaerobic resistance training. Whether there is any benefit to adult hippocampal neurogenesis in these other forms of exercise is not clear. If I'm reading this correctly, it looked like it moved the needle a little bit, but not enough to be statistically significant in their study, and definitely not as much as the aerobic exercise. Now, one advantage that scientists studying mice have over scientists studying people is that mice can be inbred for generation after generation without anyone complaining, and they did so with these mice. And of course, scientists do things like this with mice all the time, producing different strains. And there were a couple of different strains of mice that were tested in this study, some of whom were high responders to exercise and some of whom were low responders. Basically, if you gave two mice from these two separate groups the exact same workout regimen, one of them would gain a lot more muscle than the other would, and that's just a factor of genetics from the selective inbreeding. But it turns out that the high responder mice, the mice that have been bred to respond to exercise well, and coincidentally, these are the mice that seem to enjoy exercise more. They're the ones that'll go running in their mice wheel for long periods of time, things like that. Those mice who are more genetically inclined to responding well to exercise, their brains also responded better to the aerobic exercise insofar as the adult hippocampal neurogenesis. The more sedentary, more exercise-averse mice, their brains also saw benefits from aerobic exercise, but not to the same extent as their more exercise-prone peers. However, I would caution to say, don't take this too seriously, this part of it, if you're a human and you don't like exercise that much. Chances are, even if you're not a big exerciser, even if you come from a sedentary family, chances are you are not significantly inbred, and certainly not to the extent that these mice were. So it's not a question of some benefit versus no benefit, it's just a question of how much. And as we talked about last week, there are lots of other benefits to cognition besides this one particular marker of adult hippocampal neurogenesis. So just because aerobic exercise seems to be the winner in this area does not by any means discount the cognitive benefits of some of the more intense forms of exercise also on offer.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, that was all. Episode number 126, now signing off. Thank you for hanging around until the very end. Hopefully you learned lots and lots about Bacopa Monnieri. If you enjoyed this episode, please tell a couple of friends, spread it around on the internet, sprinkle it on social media, whatever you deem appropriate. All of that's very much appreciated. It is super fun hearing about people that have just found the show and are going back and listening to the early episodes. So thank you to those of you who are evangelizing on our behalf. The links to everything that we talked about here in this episode will be up at smartdrugsmarts.com/126. 126. And last week's episode, surprise, surprise, is at smartdrugsmarts.com/125. If you missed that one, that was an episode interviewing Sal DiStefano about exercise and cognition. Looking ahead to next week, I can't say that this is going to be the focus of the entire episode, but it is coming up on Mother's Day, and we will definitely do something motherly appropriate. Moms have a huge impact on the cognitive health of their offspring, and not just genetically. So if you are a mom-to-be, or even somebody that plans on producing babies with the use of a mom, then you will definitely find something worth listening to in next week's episode. That'll be next Friday as usual. Same time, same podcast, and with that same unflagging commitment to helping you fine-tune the performance of your brain and the brains of future generations. So have a great week until then, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.